Noir Plasma Chrome Bleed. Unlocking knowledge, inspiring innovation, empowering curious minds.
This website makes learning so easy. I found all my resources here.
Like what you share.
The modification of living genomes through artificial genetic engineering relies on the evolutionary conservation and universality of the genetic code. A critical scientific distinction must be maintained between the broad class of genetically modified organisms and the specific subset of transgenic organisms. The term GMO encompasses any organism with an engineered genetic modification, which includes cisgenesis, gene knockdown or knockout, agrobacterium-mediated transfer. Type I4 secretion system delivers single-stranded T-strand protected by bacterial chaperone proteins. The principal biomechanical mechanism is pronuclear microinjection, or PNI. This involves the physical delivery of linearized DNA directly into zygotic pronuclei. Replication-deficient retroviruses, lentiviruses, or adenoviruses insert genetic payloads via cell entry pathways. Host-derived phenolic signals such as acetylserine released from wounded tissues activate the bacterial VIR-A/VIR-G two-component signal transduction system. The VIR-D1/VIR-D2 endonuclease complex nicks the double-stranded plasmid at the borders, generating a single-stranded DNA molecule.
Cotton is highly susceptible to devastation by lepidopteran pests, such as the cotton bollworm.
The explants are ready for the antibiotic selection phase.
I've prepared the medium according to the protocol.
तो मंजहा सबे बनियल कथिन है उंजपिंग पंड्रन पर में पच्चमिन है आदत चरक आप चातिनन जैसे एक मंटीच में कम तचाचा है
Optimized temperature regimes. 25 degrees to 28 degrees Celsius is the optimal range for agrobacterium attachment and tDNA transfer during the initial co-cultivation period.
The roots look strong and are ready for the transfer.
Excellent. Let's get these moved over to the hardening trays immediately.
toxicity fall. Fine.
The cascade of toxicity follows a step-by-step molecular pathway. Secondary receptor interaction. The release of Cry1ac proteins into the soil through root exudation and decaying crop residues introduces distinct soil interaction dynamics. Mulching with Bt cotton residues can reduce soil quality by increasing soil bulk density and reducing nitrogen, lowering the yield of subsequent winter crops.
The refuge layout is crucial. By planting these non-beet crops right alongside the cotton, we disrupt the resistance cycle of the lepidopteran pests.
Option A, planting 25 hectares of non-Bt cotton for every 100 hectares of beet cotton, allowing the refuge area to be treated with
Transgenic mice are essential models for studying mammalian gene function, validating therapeutic drugs, and modeling human genetic diseases. Somatic progression. Understanding mammalian development is critical for timing gene transfer techniques. Upon fertilization, the single-cell zygote contains two distinct pronuclei, a larger male pronucleus and a smaller female pronucleus.
The zygote undergoes cleavage divisions, progressing through 2-cell, 4-cell, and 8-cell stages to form a compacted multicellular ball known as the morula.
The comparative biomechanical workflows involve pronuclear microinjection followed by oviductal transfer, or electroporation of vectors into embryonic stem cells. The pronuclear microinjection process relies on direct physical delivery. Zygotes are harvested from the oviducts approximately 0.5 days post-coitum. Trillis. The VMS technique utilizes efficient vibration to pierce the cell membrane with minimal distortion, enhancing zygote.
The PNI targeting efficiency is significantly higher here.
Yes, but the ESC data shows less variability across the replicates.
True, we should investigate that difference further.
First, we carefully plate the PMEF feeder layer into the dish to support the embryonic stem cells.
The pipettes are loaded. Are the parameters set?
Yes, calibrating the pressure now.
This visualization demonstrates the 5x FAD transgenic murine model, critical for studying genetic factors in Alzheimer's disease. The transgenes incorporate 5 familial Alzheimer's disease mutations, APP Swedish, Florida, and London mutations, and PSEN1 mutations.
All right, place the mouse at the center platform. We are recording the latency to first movement now.
With this patent secured, we're looking at a new model for academic research.
Indeed, the terms are favorable for both institutions.
The visualization tracks the oocyte from the ovary into the infundibulum, detailing the physiological path through the oviduct where albumin deposition begins. For germline modification, primordial germ cells are isolated from the embryonic blood, transfected in vitro using adenoviral or lentiviral vectors.
Analyzing the yield data from the automated facility.
The concentration levels look promising for the antibody isolation.
Nutritional biofortification, golden rice. In developing regions, monodiets based on polished rice can lead to severe vitamin A deficiency.
We must clarify this clause regarding informed consent. Agreed. I will add a reference to the latest EU guidelines here.
Like what you share.
Noir plasma chrome bleed. Unlocking knowledge, inspiring innovation, empowering curious minds.